and welcome to today's PHO Microbiology Rounds presentation on testing updates for the Mycobacterium tuberculosis complex, whole genome sequencing. My name is Dr. Vanessa Tran, and I am a clinical microbiologist at Public Health Ontario, and I have the absolute pleasure today of moderating today's session. We would first like to begin to acknowledge that the land that we are gathered on is the traditional territory of many nations, including the Mississaugas of the Credit River, the Anishinaabe, the Chippewa, and the Haudenosaunee, and the Wendat people, and is now home to many diverse First Nations, Inuit, and Métis people. Toronto is home to Indigenous peoples from across Turtle Island, and we are so thankful for their hospitality that allows us to live and work today. Before we begin today's presentation, I would like to start by disclosing that I have the following conflicts of interest to declare. I'm on the board of the Canadian College of Microbiologists and the Canadian Association for Clinical Microbiology and Infectious Diseases. I'm also a co-investigator or collaborator on grants funded by CIHR, PHAC, and Gilead. It is now my sincerest pleasure to introduce the speaker for today's presentation, Dr. Angela Ma. So Dr. Angela Ma is a clinical microbiologist at Public Health Ontario, overseeing laboratory testing for microbacteriology. She holds a PhD in molecular pathology from the University of Alberta and is board certified by the American Board of Medical Microbiology and the Canadian College of Microbiologists. In addition to her role at PHO, she is an assistant professor in the Department of Laboratory Medicine and Pathobiology at the University of Toronto. And with that, I will hand it over to Angela.
Thank you so much for your introduction, Vanessa. I'm very happy to be giving this presentation to everyone today for PHO Microbiology Rounds. Before I begin, I just want to disclose that I have no commercial disclosures, and like Vanessa mentioned, I do hold an assistant professor position at the University of Toronto. For today's session, the learning objectives are to explain the whole genome sequencing tested methodology for the Mycobacterium tuberculosis complex, or the MTBC, at Public Health Ontario. Also, to describe considerations for testing and interpretation of WGS results for predicting antimicrobial resistance. And lastly, to identify the role of WGS in determining genetic relatedness for MTBC for public health surveillance. So to set the stage with tuberculosis in Canada and the province in Ontario, what's shown here is 2023 data from the Public Health Agency of Canada. It shows that in 2023, just over 2,000 TB cases, that meaning active TB, were reported for the country, correlating with an incidence of 5.5 TB cases per 100,000 population. As you can see here on the graph, this trend has continued to rise over the last decade, with a small dip during the COVID-19 pandemic years in 2020. When we look at the demographics of those affected by TB in Canada, you can see that 79% of TB cases are in those that are born outside of Canada, whereas among the individuals that have TB within Canada, over 3/4 of those are affecting the Indigenous populations. When we look at TB cases in Ontario, this data that's published in January of this year by Public Health Ontario's Epidemiology Summary for Tuberculosis shows the quarterly incidence rates of TB from 2019 up to Q2 of 2024. We can see that over the last 5 years, the trend has continued to increase. And among the different regions in Ontario, the Toronto region continues to have the highest quarterly rates of TB across the province. So the Mycobacterium tuberculosis complex is the causative agents of tuberculosis disease, and what sets them apart from other non-tuberculous mycobacteria is that they're not actually known to replicate in the environment. They mainly replicate in the tissue of humans and other warm-blooded animals, and those variants are known as animal adapted. They are strictly aerobic, non-motile, very delicate, slightly curved, or sometimes straight bacilli, and they possess a very unique cell wall that makes them very resistant to chemicals and other types of disinfections and elimination, but it also gives them a number of different properties that are utilized for detection and identification in the laboratory. Their unique cell wall is rich in glycolipids, mycolic acid, and other long-chain polysaccharides. In these two images below, you can see two of the mainstay methods for microscopic detection of MTBC. In this fluorescent stain, which is the auramine rhodamine stain, this represents the AFB smear that is commonly reported in our PHO reports, and this is performed on the processed concentrate of the primary specimen. With the AFB fluorescent stain, the bacilli themselves will typically have a bright yellowish green stained appearance, and that is the mycolic acid that's being stained in their cell wall. With the Kenyan smear on the image on the right, you are seeing Mycobacterium tuberculosis complex that has already grown out in culture that is then stained and visualized with light microscopy. In this particular image here, you're seeing a very distinctive serpentine cording that can be seen with MTBC and some other non-tuberculous mycobacteria as well. And this also gives hints towards the identification tests that are then performed in the laboratory, and is also a representation of the virulence factors that the organism has, as cording factor is also involved in granuloma formation, evasion of macrophages for ingestion, and also for colony morphology. So the Mycobacterium tuberculosis complex is divided into a number of corresponding lineages if they have been designated, and a number of different variant names. So Mycobacterium tuberculosis complex did have a nomenclature change in the last few years. They are no longer referred to as individual species, and they are now referred to as variants of the main genus and species Mycobacterium tuberculosis. Lineages 1 through 9 are human-adapted Mycobacterium tuberculosis, and lineages 1 through 4 and 7 to 8 are known as Mycobacterium tuberculosis sensu stricto, whereas lineages 5 and 6, which are human-adapted, are typically associated with the West African region. For our animal-adapted variants, you can see that a number of the names do represent the type of animals that they typically are associated with. For those with zoology interest, you may recognize some of these animals, including cattle, goats, African apes, wood mice, voles, seals, etc. And typically there has been very limited transmission to humans from these animal adapted variants, but it is possible. Before moving on to talking about the details of our new test for whole genome sequencing, I think it's important to orient the audience to the testing process that occurs for AFB testing or mycobacteriology testing at PHO. So I'm going to be walking through the life cycle of a specimen that comes in through the lab and the various different stages that the audience may see our reporting results. So a specimen that comes in will go through a series of processing stages to prepare the specimen and remove normal microflora that could otherwise outcompete AFB from growing in culture. The fluorescent stain is then performed, and then the specimen is inoculated into both solid and liquid media. The liquid media is typically referred to as the midget, which stands for the Mycobacterial Growth Indicator Tube, and the solid media is typically a Lowenstein-Jensen slant. Depending on the smear grading or if approval is given, we will then perform our TB-PCR, which is a molecular assay that detects TB-DNA and also predicts resistance to rifampin and isoniazid. Cultures will then be incubated for a total of 7 weeks before being reported as no growth. And if they are positive, they will then be stained with the Kenyan, and then a number of different identification tests will be performed. For non-tuberculous mycobacteria, there are a separate number of tests that are done for those. And in these images here, you can actually see a non-tuberculous mycobacteria growing in that liquid medium as very small yellow flecks of colonies and a corresponding Kenyan smear. For our MTBC, what you can actually see here again is the cording phenomena seen in the Kenyan, and also these are solid media LJ slants of Bovis BCG. For isolates that are identified as MTBC, they will undergo first-line PDST and our new test whole genome sequencing. Depending on the susceptibility profile and the results from first-line PDST, they will then either be sent to the NML and have second-line testing performed, or it will stop if they are pan-susceptible. So Because we are going to be talking about a number of different tests that can generate resistance results, I did want to touch on the two tests that we already have that are routinely performed at PHO. That includes our TB-PCR. So this is a new test that we shifted to in the last July in 2024. We are now performing the BD-Max MDR-TB assay. And all of the test information for this test is available on PHO's test index. This is performed on concentrates and not primary specimens, with the exception of CSF and formalin-fixed paraffin-embedded tissues. They are routinely performed on AFB smear-positive specimens from new untreated patients and those who have not had a history of a positive AFB culture within the last 3 years. And like I mentioned before, smear negative requests do need approval from the microbiologist for them to be tested. For PDST, Ontario uses the BACTEC Midget 960 method that is based on critical concentration. So critical concentration means the concentration of drug that inhibits 95% of wild-type MTBC that does not have resistance to that drug in question. For all Ontario TB cases at PHO, we perform first and second line PDST here. For MDR strains, those that are multi-drug resistant and have resistance to rifampin and isoniazid, they are reflexively forwarded to the National Microbiology Laboratory in Winnipeg, Manitoba for bedaquiline and clofazimine testing. As we move to talking about the testing details about our new MTBC WGS test, all of the test details for both PDST and whole genome sequencing are available on our Mycobacterium TB susceptibility test information page in our PHO test index. So whole genome sequencing is a molecular sequencing method that looks into the makeup of an organism by reading most or all of its genetic code. In the context of whole genome sequencing for MTBC, the organism possesses a DNA-based genome that is approximately 4.4 million base pairs in length, and with whole genome sequencing, we are able to determine approximately 90% greater of its genome. So about the clinical test that we have just launched in February of this year, whole genome sequencing of MTBC is not a standalone orderable test, and requisitions that come with that order will have it rejected. It is routinely performed on the first culture of MTBC isolated for a patient at the same time that PDST is also ordered on that. And for both PDST and whole genome sequencing, they're repeated at 3 months if cultures remain positive for a patient. Whole genome sequencing is performed weekly at PHO's laboratory in Toronto, and the turnaround time is up to 21 calendar days from the date of culture identification report to account for possibly lowly replicating or low metabolically active strains of TB or those that struggle to grow in liquid and solid culture. For the most part, our turnaround time is less than 21 days for whole genome sequencing of MTBC. So this test is performed on midget subcultures of MTB isolates, midget subcultures being the liquid media. Sequencing involves library preparation and short read sequencing using the Illumina platform, and then the generated sequences are then processed and analyzed using a laboratory-developed pipeline here at PHO. To walk the group through the process of that bioinformatic sequencing and pipeline, what essentially happens is that the data in the form of raw sequencing reads are then analyzed through this pipeline, starting with a number of different quality checks and trimming first. Then a number of bioinformatic tools are then used to provide results for taxonomic classification and metagenomic analysis. Following that, there are a number of steps that happen, including dehosting of contaminating reads, i.e. removing human DNA reads out of the sequencing, and then aligning the remaining reads that are in high quality to a reference genome for MTBC. In this case, we are using H37RV. This is a very well-characterized Mycobacterium tuberculosis genome, does not have resistance mutations, but it is actually not reflective of current circulating TB strains in Ontario. Next, additioning filtering and bioinformatic tools are then used to determine the complex member and the lineage of that sample, and then also to provide the genotypic AMR predictions. So this is essentially the life cycle of the bioinformatic analysis for the clinical portion of this whole genome sequencing test. For our surveillance for TB for public health units, we also do perform additional sequencing analyses using SNP calling pairwise analysis to determine the genetic relatedness between strains. For the clinical whole genome sequencing test, there are three major test components. That includes the complex member identification, the complex lineage determination, and also the AMR predictions. Specifically for the AMR predictions, there are a number of different considerations here. Routinely, the test will report the first-line anti-TB drugs resistance predictions, including isoniazid, rifampin, ethambutol, and pyrazinamide. Otherwise, I will be referring them to as RIPE, and for moxifloxacin as well, because we recognize that the fluoroquinolones are the most commonly requested second-line agent for PDST. If in any circumstance there is resistance predicted to any of these 5 drugs by whole genome sequencing, we will reflexively be reporting out AMR predictions for second-line drugs that are analyzed by this test, including amikacin, capreomycin, ethionamide, kanamycin, linezolid, levofloxacin, streptomycin, clofazimine, bedaquiline, and delaminid. In addition to release of our second-line AMR predictions, we will also be routinely reflexively doing additional PDST depending on certain AMR predictions that are detected. If rifampin resistance is predicted, then PHO will reflexively perform a full panel for second-line PDST. In a setting where isoniazid resistance is predicted in the absence of rifampin resistance being predicted, then we will be performing high-level isoniazid and moxifloxacin PDST. If moxifloxacin resistance is predicted by whole genome sequencing, then PHO will also be performing confirmatory moxifloxacin PDST. If there is a setting in which there is no resistance predicted by WGS, and there is possibly intolerances or side effects occurring with anti-TB therapy, second-line AMR predictions can be released on approval from a PHO microbiologist, and physicians are recommended to be contacting PHO's customer service center so that they can have a consultation with the PHO microbiologist. So in this table here, we are now showing all of the different antimicrobial agents that are targeted by PHO's WGS assay and their corresponding genes analyzed. So the target genes and the resistance predictions are based on only high confidence mutations associated with resistance that are included in 2023 edition of the World Health Organization's Catalog of Mutations in MTBC and their association with drug resistance. With a test as complex as whole genome sequencing that can be used to guide clinical decision making, the validation is a critical piece. And with something that where there are PDST results that are available for comparison, diagnostic sensitivity and specificity is a critical part that is evaluated in our validation. What sets whole genome sequencing apart from other molecular or lower complexity tests typically for validation is that what is defined as diagnostic sensitivity and specificity is a little bit different, and I'll be introducing that to our audience here. So with a microbial whole genome sequencing assay, diagnostic sensitivity refers to its ability to accurately detect any sequence variation, whether it's known or unknown, within the genomic regions analyzed within the assay. And two parameters that are evaluated for diagnostic sensitivity is the positive percent agreement and a positive predictive value. For diagnostic specificity, that refers to the ability of the test to not detect sequence variation within the analyzed genomic regions when there is none present. And for that, we look at negative percent agreement and negative predictive value. And for each of these 4 different parameters, there are a number of calculations that look at the true positive, true negative, false positive, and false negative results for that calculation. In the setting of whole genome sequencing for MTBC, because PDST is still regarded as the gold standard method, we are able to compare and evaluate its performance by looking at our PDST results. In this table here, you can see the various different parameters that are assessed for diagnostic sensitivity and specificity with our WGS genomic AMR predictions when compared to PDST. That includes for our first-line agents and a number of our second-line agents. Overall, you can see that there is very high agreement for the majority of our drugs, and the performance of PHO's MTBC WGS assay is comparable to those that are also performed in Canada, being British Columbia and at the National Reference Laboratory, the NML, in Manitoba. Overall, for agents where there is lower diagnostic sensitivity and specificity, reasons for that include circulating MTBC isolates that have vary in the different geographic regions and particular certain resistance mutations that are not reflected in the global WHO catalog that is used as our backbone for AMR predictions. In this table here, you can also see that there is no results for comparison specifically for clofazimine and bedaquiline, and that is because of insufficient samples, as there's very low rates of MDR-TB in Ontario, luckily, and there have been no critical concentrations formally established by the WHO for these two drugs yet. So with our genomic AMR predictions, it is important to recognize that they are presumptive as they do not provide direct evidence for resistance. Additionally, while first-line agents and the resistance mutations associated with them have been very well researched, the second-line agents are relatively lacking, but there's a number of literature and research that continues to grow in that area. And additional updates to catalogs and databases will allow for more high-confidence mutations to be included in different catalogs and in pipelines over time. Also, the absence of mutations by genotypic AMR predictions does not exclude the possibility of other contributory mechanisms of resistance. After all, not all resistance determined phenotypically can be captured by genetic changes in the specific genes examined in this assay due to the complexity of how antimicrobial resistance develops. Lastly, PHO will not be stopping PDST. We will be continuing to use PDST as it's together with whole genome sequencing that really the most comprehensive picture and resistance profile for an isolate can be determined. And lastly, one of the major benefits of genotypic AMR predictions and utilizing whole genome sequencing is that the tools and the databases used are dynamic. They continually are updated by different groups and consortiums, and that allows for whole genome sequencing of MTBC to remain current and up to date as well. So overall, this is a summary of the whole genome sequencing for MTBC clinical assay. Like I mentioned before, it has three main test components being complex member, lineage, and AMR predictions. In the middle column here, you can see all of the possible results that individuals may be seeing in their PHO reports and certain test comments that are applicable to certain results. To help the audience digest this further, I'll then be moving on and presenting a couple of example reports and walking the audience through them. So in this first example here, this is a scenario of an MTBC isolate where no resistance is predicted by AMR predictions with whole genome sequencing for our first-line agents and moxifloxacin. With all of our mycobacteriology testing at PHO, all of our test reports will always have the test listed in the same order. They will always start with the AFB smear that is performed on the processed concentrate, followed by the mycobacterial culture results. In this example here, you can see that it was identified as Mycobacterium tuberculosis complex. And then below that, you will then see the PDST results. In this box in green, this captures all of the new MTBC WGS results, including complex member identification with the test method in parentheses, here saying WGS. Similarly with complex lineage, it also shows WGS, and then it will have individual lined entries for the genotypic AMR predictions and the appropriate test comments that come with the test. In this example here, you can see that there's no high confidence resistance mutations detected by whole genome sequencing and that resistance cannot be ruled out. In this second example here, this is one that has resistance predicted by whole genome sequencing to moxifloxacin. Again, the various AFB tests are listed in the same order. And to direct your attention to this line here in this purple box, you can see that moxifloxacin has a result of resistance predicted in genes analyzed, and those specific genes being JAR-B and JAR-A. Automatically in the PHO report, you will be seeing the resistance predictions for the second line agents also reported in the second purple box here as well. Not shown in these example reports is if in the scenario a specimen is tested with our TB-PCR assay, and in those situations, what you'll be seeing is that the Real-time PCR results for the BD Max will be showing up after the PDST results, but before the whole genome sequencing results. In this next session of our presentation, I'll now be talking about some of the considerations for TB-PCR, PDST, and whole genome sequencing, as there are now a number of different situations and instances where individuals may now be seeing different susceptibility and resistance results for MTBC. So this is again the overview of AFB testing here at PHO. And in these three different circles, you can now see that there are a number of different areas where you may be reading some type of TB detection or resistance prediction and susceptibility testing result. So again, there are molecular tests that are being performed at PHO that not only detect MTBC DNA, but also provide resistance predictions for rifampin and isoniazid. That can include the TB-PCR, which is the BD Max MDR-TB assay that's performed with specimen concentrates, or FFPE, or on positive cultures identified as MTBC, which is done using DR plus line probe assay, that is only performed on request for PHUs, for public health units, to support de-isolation decision making. Of course, the gold standard method remains phenotypic drug susceptibility testing, and this is done using the Midget 960 and critical concentration method. And lastly, we have our new test, whole genome sequencing AMR predictions, that is done with whole Illumina sequencing. So the question that the audience may have is, can discordant results occur between one or more of these assays? And the short answer is yes. And in the next few minutes, I'll be going over a number of the different considerations and scenarios for why this can happen, and also providing some examples of this as well. So before we talk about specific considerations for whole genome sequencing genotypic AMR predictions, I did want to dedicate some time to talk about false positive and false negative scenarios for the TB-PCR, because what makes this one different from the other methods, being PDST and whole genome sequencing, is that this is performed on the primary specimen concentrate, and as a result, there are a number of additional considerations that can impact the results that may not be seen with testing of the culture. So these considerations are broadly applicable to all PCR-based methods for MTBC detection and resistance prediction, and are not limited to a specific assay. I think a critical point for TB-PCR is that the analytical sensitivity is directly associated with the smear grading, or whether or not there is AFB present, and the burden available in the specimen and sample being tested. In situations where there's low smear grading or no AFB seen, that will correlate with lower sensitivity in PCR detection, and sampling or testing variation will be impactful even more so in low smear grading or no AFB seen scenarios. It's well known that medications, mouthwashes, and rinses, particularly in the case of respiratory specimens, and complex matrices such as body fluids, CSF, and FFPE that can be pleocytic, can lower sensitivity or possibly cause false positive and false negative situations. So these various agents and their considerations can cause non-specific fluorescence or cross-reactivity with primers and probes, leading to false positive results. But in situations where there is a high number of possibly proteins or white blood cells which release very high amounts of DNA, it can also cause competition in primer and probe binding, leading to false negative results. Additionally, high bacterial loads of MTBC, in addition to non-tuberculous mycobacteria and other microorganisms in the specimens, can not only cause false positive MTBC detection results, but can also cause false positive resistance prediction as well. It has been documented with the expert that in cases of very high burdens of MTBC that are not resistant to rifampin, they have actually caused false resistance prediction results for rifampin, because there's just so much overwhelming MTBC DNA, and that results in altered target hybridization and amplification. Additionally, NTMs and other microorganisms that can be present in a specimen can cross-react due to sequence homology to MTBC or rifampin and isoniazid targets, and also cause primer competition and cross-reactivity there. Specifically for whole genome sequencing and genotypic AMR predictions, it's very well known that whole genome sequencing offers the benefits of being able to detect borderline resistance mutations for rifampin and ethambutol that at times cannot be detected by PDST. And this is because these resistance mutations confer resistance near the critical concentration for PDST, but they don't actually pass the threshold to be triggered as phenotypically resistant by the test method. However, these mutations may be associated with in vivo resistance and treatment failure. Pyrazinamide or PZA has always been a challenge for PDST. Pyrazinamide is actually the prodrug form of the active pyrazinoic acid that is actually the active drug. However, this activation requires an acidic environment, and for PDST testing, that acidic environment is actually initiated by changing the pH of the liquid media. However, this actually inhibits MTB replication in culture, as that low pH is suboptimal for organism replication, and that can at times make PDST for PZA very challenging, and in less experienced laboratories can actually result in inaccurate PZA PDST. On the whole genome sequencing side, the PNCA gene, which is the target for PZA resistance prediction, does have the challenge that there is actually no hot spot within the gene where resistance mutations commonly arise. Resistance mutations can span the entire length of the gene, meaning that low frequencies of genes may result in them not being included in those global catalogs that document high confidence resistance mutations for PNCA. For isoniazid and fluoroquinolones, which have both low-level and high-level PDST routinely performed on them if low-level resistance is detected, whole genome sequencing actually has very high agreement with low and high level resistance that is seen with PDST. The mechanism behind low-level phenotypic resistance for these two drugs is that multiple genetically linked resistance mutations are additive to confer high-level resistance in the setting of the INHA gene for isoniazid and the JAR-A and JAR-B genes for the fluoroquinolones. In settings where a mutation in the CAT-G gene is detected for isoniazid, that typically will always confer high-level resistance to isoniazid. Now, then a question that the audience members may have is that why is there no resistance predictions available for pretominid, and why isn't pretominid PDST more widely available? And one of the challenges associated with this is that there's no critical concentration that has been established by the WHO yet due to different intrinsic susceptibilities to the drug across different lineages of MTBC. So lineage 1 isolates have intrinsically higher pretominid MICs or minimum inhibitory concentrations than lineages 2, 3, 4, and 7. Additionally, while delaminid and pretominid are both nitroimidazoles, there is actually not always cross resistance that can be conferred if resistance is detected for delaminid with pretominid. I'll now be walking the audience through a number of different examples of scenarios where you may experience or encounter conflicting or discordant results from some of the different types of AMR testing that can be done for MTBC now. So in this first example here, this is a smear negative specimen that eventually grew out as Mycobacterium tuberculosis complex. Here, the DR plus Lyme probe assay was performed on request for the public health unit to support their de-isolation decision making. We can see that from the positive culture of TB, it was again able to detect DNA for MTBC and also resistant to isoniazid. However, it was not reportable for resistance predictions for rifampin. When PDST was performed, it confirmed the isoniazid resistance, and it reported that rifampin resistance was not detected and it was susceptible. Now for a new drug, with our new test for whole genome sequencing, we're now able to have another layer of deeper investigation onto genetic resistance predictions. We can see here, not only did it provide a complex and lineage identification, but it also again confirmed the isoniazid resistance. But in this instance, it was actually able to predict rifampin resistance in the gene analyzed. And behind this is actually the detection of one of those WHO-endorsed borderline resistance mutations in the RPOB gene that confers phenotypic resistance to rifampin, but is below the critical concentration for PDST at 1 milligram per liter. In the second example here, this is really to illustrate the additional investigation and details and data that can be obtained now of whole genome sequencing in situations where a false positive for TB-PCR may be encountered. So in this example, we have a 2+ AFB specimen that eventually did grow out TB. On that 2+ AFB specimen, the TB-PCR was ran and isoniazid resistance was detected. When it grew out in culture, PDST showed that it was susceptible to low and high level isoniazid, and whole genome sequencing was able to determine that there was no high confidence resistance mutations detected. Prior to whole genome sequencing, troubleshooting to determine whether or not that false positive was actually the case for that TB-PCR would have been much more challenging. However, now with whole genome sequencing and some of the additional bioinformatic tools that are utilized in that pipeline, it now gives us at PHO a deeper idea into seeing what is actually going on in that sample. With whole genome sequencing, the metagenomic analysis for this case actually showed that the sample had high amounts of Raphia mucilaginosa and Streptococcus mitis DNA present. And it's possible that both of these organisms, or one of them, may have cross-reacted during the TB-PCR assay that's performed on the primary specimen concentrate. So both Raphia and Streptococcus are normal microflora of the upper respiratory system, and because they are actually even detected as DNA reads in the subculture of a positive MTBC culture, it is very likely that they were in much higher abundance in the primary specimen, and that could have contributed to cross-reactivity and a false positive isoniazid resistance result on the TB-PCR. So in summary, again, I'd like to emphasize that the database for resistance-associated mutations contains only high-confidence mutations from a curated global catalog, and that the overall agreement between whole genome sequencing genotypic AMR predictions with PDST is very high. Additionally, in any situation where the genotypic AMR predictions from whole genome sequencing do not agree with a PDST result, please be reassured that these would only be reported after the PHO microbiologist has reviewed and approved its release, meaning that there is evidence and justification behind the discordance, and we would not be reporting it otherwise. Overall, some take-home messages on how to interpret WGS results within the context of PDST results. If whole genome sequencing does not predict resistance, but PDST is reported as resistant for a specific drug, then remember that there are always cellular, protein folding mechanisms, or uncharacterized genotypic mutations that can cause resistance, and to follow those PDST results. And this is particularly in the case for PZA and ethionamide if second-line testing is being done. If MTBC WGS predicts resistance, but PDST is reported as susceptible, then there is very strong evidence behind the AMR prediction to support that drug resistance that is not possibly detected by PDST can be associated with in vivo resistance and treatment failure. Overall, whole genome sequencing and genotypic AMR predictions provides a number of advantages now. It allows for the prediction of resistance-associated mutations that may contribute to treatment failure that is not detectable by PDST, as illustrated with our example of that borderline rifampin resistance isolate. Additionally, it does offer faster turnaround times for not only first-line drugs in situations where there is perhaps low metabolic rates or challenges in replication in liquid media, but particularly also for second-line drugs as well. Additionally, with the launch of our test, we are seeing that our turnaround time has been able to provide earlier AMR predictions for our first-line drugs when PDST is still pending. Additionally, because AMR predictions are being performed for both first and second line drugs, if in situations where a pan-susceptible isolate um, is being present and causing TB disease in an individual, but they are experiencing intolerances or side effects and second line drugs are necessary, release of AMR predictions for second line agents are available faster because second line PDST will need to be set up subsequently. Another benefit to genotypic AMR predictions is that next generation anti-TB drugs that may not have PDST methods available for testing are available through WGS, and these results also enable physicians to apply for access to these next generation drugs from the TB Consilium. Lastly, genotypic AMR predictions allow for the detection of heteroresistance, meaning those where there are subpopulations of susceptible and resistant MTBC present in a patient to support clinical decision making, and in cases where mixed cultures hinder PDST, so that's not possible, AMR predictions may be possible to be provided to help guide treatment decisions. In this last section of our presentation, I do want us to devote some time to talking about the utility of whole genome sequencing now for public health surveillance and supporting TB surveillance in the province of Ontario in the context of genomic epidemiology. So, Public Health Ontario has historically been the only province in Canada that provides a digital interactive TB surveillance portal for our public health units in the format of OutTB. So what we see here is our existing digital interactive tool that utilizes PCR-based genotyping methods to link um, laboratory data with TB tuberculosis case data to support contact tracing and investigations. It allows for line list searching for identical related cases and also to provide a number of additional laboratory contextual results as well. With whole genome sequencing, we will now be providing an entirely new tool in the spring of this year to support our public health units, known as OTB by WGS. This is an enhanced tool that now integrates whole genome sequencing and single nucleotide polymorphism distances for more precise identification of genetic links between cases, and it also provides a number of additional new features such as data filtering, weekly updates, and updated definitions to the Canadian tuberculosis standards with the current edition. In this snapshot to the left, you can actually see some of those additional new filters available for our public health unit users and also our case searching functionalities as well. A table will be provided that shows all of the laboratory confirmed culture positive tuberculosis cases, and it's interactive so that users can either search for a case or they can click a case, which will populate a secondary table that shows all of the genetic relatedness results by whole genome sequencing to that target case. In this example here, when that case was selected, we could see that there were 6 matching or related cases by whole genome sequencing that are identified in our database. And you can see that the degree of genetic relatedness is also indicated in this match type column. For a more drilled down look for each of these clusters, we will also now be providing SNP distance matrices for each of the selected cases so that the individual relationships in a pairwise manner between cases can be analyzed for our public health units. As we conclude this presentation, some summarizing notes are that whole genome sequencing for MTBC can be used to support clinical decision-making for adjustments to therapy together with PDST results to really provide the most comprehensive resistance and susceptibility profile. Secondly, whole genome sequencing genotypic AMR predictions are based on a very strict catalog that only includes high confidence resistance mutations. And lastly, whole genome sequencing provides much greater accuracy and resolution compared to genotyping in identifying genetically related MTBC isolates to support TB surveillance, contact tracing, and investigations. As I end today's presentation, I would like to give a very large thank you to the group behind developing this test, both on the clinical side, the pipeline, and also for our surveillance aspect. This is very much a labor of love from PHO, from a very large group of individuals. And as I end things, I do want to let the audience know that on March 24th, it is World TB Day, and there will be more additional PHO rounds and sessions for tuberculosis that will be coming in the near future. For more information about this test, I highly recommend that our audience contact PHO's Customer Service Center, either in their telephones, which can then be routed to myself or the Tuberculosis Laboratory or the Microbiology Laboratory at PHO to appropriately answer your questions, concerns, or consultations. Lastly, here are my references for this presentation, and thank you very much, and happy to move on to the Q&A session.
Thank you so much, Angela, for that very insightful and very comprehensive presentation. We will now move on to the Q&A segment to answer questions from the live audience. As a quick reminder to everyone, if you have not already, please enter your questions into the Q&A pod below. And we have a couple of questions already coming in. So just give me one sec. I'm going to try to group them as best as I can by topic. And maybe I will first start with some of the reporting. So some of the examples that you showed could look quite complicated. And so there's a question about on the report if there would be guidance or comments on how to interpret the lineage result and other results.
So that's a really good question. For our lineage and complex results, the information is always available on our test information page on the PHO test index. So there is a detailed table that documents all of the corresponding complex members and their lineages on the Mycobacterium TB susceptibility webpage for all of our reports as well. For the lineage results, they will always have the numerical value, whether that's lineages 1 through 9, or if they're animal adapted, then they will have their specific designations there. And it'll also provide a geographical context of how these lineages are commonly described in literature, and are actually a reflection of how TB has evolved over time, because they can be linked very distinctively to certain geographical regions. But all of that information is available on PHO's test information index.
Thanks, Angela. I'm going to try to combine the next 4 questions together because I think they're related to how the cases are linked and the SNP distances. So I guess the overall general question is, how is it used to link cases together? I don't know if it would help to provide a very small example. And in doing so with the SNP analysis, what is the distance threshold for a match? And when you present the SNP distance matrix, will there be any sort of guidelines in terms of how to interpret those SNP differences on the report?
Yeah, so that's a great question. So there are two main methods for determining genetic relatedness for TB. One of them being SNP distances, which is what's utilized in this pipeline here at Public Health Ontario. And another commonly used method is of core genome MLST, or multi-locus sequence typing. For both of these methods, what it is essentially looking at is how many alleles or single nucleotide polymorphisms are different when sequences are compared against each other. And for how that works for surveillance methods, it's when two samples and their genomes are being compared and looking at how many shared regions there are. If there is a SNP present in one sample that is not present in the other, then that would be counted as one. And then in different regions where they have the shared positions, those will be then added up, totaling. And then that can be used to determine how related a sample is. With how we have developed SNP distance calling for our tool for TB, there are thresholds for a match. For our public health users, we do have additional user guides and technical notes that will be released with our new tool that will be available for reference. And, for the shorthand answer, the cutoff for genetic relatedness is 15 SNPs for TB, and that there are different related cutoffs for whether they are considered matching for 0 to 3, closely related 4 to 6, related will then be from up to 15 from there. And all of those definitions are available on the tool for our public health users and in those technical notes that will be distributing.
Thanks, Angela. Sorry, I'm calling you Angela. So based on your experience with the WGS reads, what proportion of them align to the reference genome? I guess in comparison to other genomes that I guess it could be some cross-reactivity to, like other respiratory genomes, like you had mentioned earlier.
Yeah, so that's a very good question. For the most case, the decontamination and dehosting steps is really meant to remove human DNA, and for the most part, human DNA does not have much sequence homology to the Mycobacterial tuberculosis complex genome. What's most challenging will be with NTMs, of course, because there is a much higher amount of sequence homology being in the same genus to the tuberculosis genome. And it really depends, and it can impact the AMR prediction results. And for those, it really depends on how much reads for this non-TB organism is present. And we really handle these on a case-by-case basis because with how we are using midget subcultures, specifically that is being extracted for whole genome sequencing, that step itself really does help to knock down non-TB reads present because we are additionally enriching for more Mycobacterial tuberculosis to grow in the subculture before extraction.
Okay, the next question is, I guess, more of an epidemiological question. And wondering if you can comment on how much MDR there are among all persons with TB in Ontario versus among First Nations.
Yeah, so that's a really good question. So overall, luckily, MDR cases are very low in both Ontario and in Canada. In national data, we can see that approximately 89% of our TB cases are pan-susceptible, meaning they have no resistance to any first-line drugs. While 11% of that proportion do have resistance to first-line drugs, that doesn't mean all of them are MDR-TB. Of MDR-TB, essentially 1.6% of isolates in Canada are MDR-TB, and in Ontario, we see a very small proportion of MDR-TB cases per year. In terms of First Nations, we don't have that data available specifically to answer that, so I can't speak to that question.
Okay, thank you. Earlier in the presentation, you mentioned that other provinces are also using whole genome sequences for MTB. Do you know how comparable the methods are and whether or not, I guess, results could be exported or compared from one jurisdiction to another?
Yeah, that's a great question. So, across Canada, Ontario and BC are the two provinces that have developed clinical AMR predictions routinely for their MTBC isolates that are identified. Manitoba, being in proximity to the National Microbiology Laboratory, does also have their isolates routinely sequenced as well for that same purpose because they have the reference lab available for them. Overall, for AMR predictions, there are two main tools for bioinformatics that are used, and there's a lot of comparability, not only in the tools that are being used across the provinces, but also in the versions of them as well. And there is a lot of dialogue and collaboration between the different provinces and territories to be exploring and, working together to evaluate our tools and how to improve them. So there is some level of agreement and comparability across them.
That's great. Um, I guess just a quick question in terms of you. So I think you did a wonderful job outlining some of the potential discordant results that you can get between the various methods. In your experience so far after going live, I guess not that long ago, how often do you see the discordance, or can you make a guess in terms of like how often you would see discordance?
That's a really good question. I think with the AMR predictions specifically, um, they are very known reasons for the discordance that we can anticipate them in settings, especially when PDST seems to be challenging. We see that a lot with the rifampin resistance, possibly also with the isoniazid or the moxifloxacin low-level resistance now. We have also seen situations where there's heteroresistance for certain drugs now, where they are taking very long in PDST, and AMR predictions actually is able to show us that there is perhaps 40% that is resistant and 60% that is susceptible. Um, causing heteroresistance. And since we've launched, we have seen a couple of cases of these. And based on our historical data, since we have looked at over 1,000 isolates for our comparison with PDST, we have been able to identify those kind of more common situations for discordance from that data.
Thank you. Okay, so it looks like there's no more questions. So thank you to everyone who submitted questions. And again, I'd just like to remind everybody that March 24th is World TB Day. Also, coincidentally, it is my birthday, so you can choose which one you want to celebrate, or you can celebrate both. So as we wrap up today's PHO round session, I would sincerely like to thank you, Dr. Angela Ma, for presenting. I would also like to thank everyone who joined us for today's event. You will receive a brief anonymous survey. Please, please, please take a moment to complete it, as it does help to enhance our future programming. And lastly, please save the date. Topic 2025 will take place on March 26th and April 2nd. Please visit www.topic.ca. That's tophc.ca for details. And thank you. Please go outside, enjoy this wonderful sunshine if you're in Toronto, and have a wonderful day.